So in this video, I'm going to review what you need to study for Quiz 5. And so the majority of Quiz 5 is all things related to staph and strep. And so that includes the Mannitol salt agar, the throat culture on blood agar, camp test, Bile Esculin agar, optogen and bacitracin sensitivity, coagulase, catalase, general characteristics of staph and strep. And then I'm adding one additional part to that, and that is the fungi, because I want you to start reviewing that now for your lab final. So I did add fungi to this list, but I'm going to walk through the slides for these and just kind of quickly review with you what is on those slides and what is important to know. And so let's walk through and take a look at this. So you have a slide that looks like this, and it's comparing and contrasting Staphylococcus and Streptococcus. And so what I did was I color-coded. If it's black, it's the same. If it's red, it's different. So in terms of shape, Staphylococcus, Streptococcus, they're the same. They are both cocci, which would be our spheres. In terms of gram reaction, both are going to be gram-positive. But where we start to get different is in the arrangement. Staphylococcus is going to be found in clusters, right? Staphylo refers to grape-like clusters. Strepto refers to chains. Where it's found in the body is going to be different. If you're talking about Staphylococcus, it's going to be found primarily on the skin and the nasal passage. If you're talking about Streptococcus, mouth, throat, vaginal area, GI tract, etc. Catalase reaction. So you might wonder, well, why was catalase on that list of tests to know? Wasn't catalase part of exam 2? And the answer is yes, but the reason that it's there is because catalase is a rapid test, key rapid test, that can be done to differentiate between Staphylococcus and Streptococcus, meaning if you were looking at both, and you wanted to quickly differentiate whether it was Staphylococcus or Streptococcus, you could run a catalase test. Because if you remember, Staphylococcus is a facultative anaerobe. It grows with or without O2. And if O2 is present, it will use O2 as its final electron acceptor. And as a result, it's going to be catalase positive. On the other end of the spectrum, if we look at Streptococcus are aero-tolerant anaerobes. They're strict fermenters. And so again, what that means is that they grow where O2 is present, but they don't use it. And so as a result, Streptococcus species would be catalase negative. They don't use O2 as their final electron acceptor, and therefore they don't have catalase. Remember that they would have peroxidase instead. So if we were trying to differentiate between staph or strep, we could quickly just run a catalase test. Get a glass slide, take a little bit of the bacteria, put it on the glass slide, add a drop of hydrogen peroxide. If it bubbles, is it staph or strep? So you want to think about that. If it bubbles, is it staph or strep? And the answer is if it bubbles, you're looking at Staphylococcus. Because Staphylococcus is catalase positive, they're going to take the hydrogen peroxide that you added to the bacteria, and they're going to produce water and O2, which is the gas that causes the bubbles. Streptococcus is not going to produce the O2 gas, and therefore it's not going to bubble. And so the reason that the catalase test is on this quiz and on this practical is because it's a way that we can differentiate between Staphylococcus and Streptococcus. Reaction to cold or dryness. So Staphylococcus, you have to remember, is found on the skin. So it's well adapted to dryness, right? It's tolerant to temperature changes. It's tolerant to dryness because it's used to being out in the open. So we would say that Staphylococcus is resistant or not sensitive to cold or dryness. Streptococcus, on the other hand, is fastidious, which means that it has very specific growth requirements. And so if you think about the types of places where you find Streptococcus, mouth, throat, vaginal area, GI tract, those are all very moist. Streptococcus is not resistant to dryness. It needs to live in a moist environment. In terms of salt tolerance, Staphylococcus is on your skin, therefore is osmotolerant. Streptococcus, on the other hand, is not osmotolerant. And so that's a difference between them as well. And so you want to know these general characteristics of staph and strep so that you can compare between the two and know how to identify which one it is. So you also had a slide that was comparing Staph epidermidis and Staph aureus. And so you want to review this as well. So Staph epidermidis, 100% of people carry it. So everybody is going to have Staph epidermidis on their skin as part of their normal flora. In the case of Staph aureus, some people will and others won't. So it's estimated that between 25 to 30% of people carry Staph aureus. If you are a medical worker, that goes up to about 50% probability that you carry it. In terms of pathogenicity, disease-causing ability, Staph epidermidis, not pathogenic, doesn't cause disease. It can be an opportunistic pathogen, meaning if you are immunocompromised, it can cause disease, but that's not very likely. Whereas for Staph aureus, it's more likely to be pathogenic. It's not always. There are different strains of Staph aureus. Some are much more pathogenic than others. Think of MRSA. Methicillin-resistant Staph aureus is very pathogenic. It causes severe disease. But not all strains of Staph aureus behave the same, but it can be pathogenic. Staph epidermidis is part of normal flora. Again, 100% of people carry it as part of their normal flora. In terms of Staph aureus, some people can be what are called transient carriers, meaning sometimes they carry it and others they don't. So 2/3 of people might carry it sometimes, meaning sometimes they'll test positive and other times they won't. So in many cases, Staph aureus is probably not part of somebody's normal flora, but it can be. Fermenter of mannitol. Mannitol is a type of sugar. You're going to see in our MSA agar, our mannitol salt agar, mannitol is going to be our differential ingredient. And that's because Staph aureus can ferment it. So notice it says yes, Staph aureus can ferment it. Staph epidermidis does not. And so that is the differential ingredient in MSA to know if we're looking at Staph aureus or Staph epidermidis. And then the next one is the coagulase reaction. And so we'll talk more about this in a minute, but for now I just put this on here. So Staph epidermidis, coagulase negative, Staph aureus, coagulase positive. And again, that comes down to virulence. Coagulase is what we call a virulence factor. It's going to cause disease. So because Staph aureus is pathogenic, it has coagulase. Staph epidermidis is generally not pathogenic, so it's going to be negative for coagulase. And so that's a test that we can do to confirm whether it's Staph aureus or Staph epidermidis. So you want to understand the different virulence factors that staph has, right? And virulence again means disease causing. And so there are 3 main ones that we have talked about. So coagulase is going to help the bacteria form a blood clot. So for Staph aureus, about 97% of Staph aureus would test for coagulase being positive. The advantage for having coagulase, what that does for the bacteria is that it forms a blood clot around the bacteria and it helps shelter it and protect it from the immune system. And so because, right, if it's in your blood, for example, or it's in a cut, the blood flowing to the area is going to send white blood cells to fight that foreign invader. If staph is contained within a clot, it's going to help protect it from those white blood cells. Beta hemolysin. This is an enzyme that's used to digest red blood cells, and it uses the contents for food and multiplies. So again, this is just allowing the bacteria to then use the blood cells, the red blood cells, for food. And then lastly, we have our staphylokinase. Staphylokinase is kind of, you can think of it the opposite of coagulase. Staphylokinase is used to dissolve the clot instead of form the clot, and that allows for spread to neighboring tissues. So when resources become scarce within that blood clot, then they're going to break down that blood clot, and the blood clot, when it breaks down, it's going to allow the bacteria to disseminate and to spread to other tissues, and then the bacteria can go back to forming coagulase and form a clot and hide out from the immune system. And so you want to have a general understanding of these virulence factors that staph can have. You don't, for my class, you don't have to understand or you don't have to memorize the differences between folliculitis, abscess, furuncle, carbuncle, etc. But you do want to know the types of diseases that are associated with staph. So skin type infections, right? So an infected hair follicle, for example, it could be systemic, might cause sepsis. Staph aureus can cause pneumonia, which is a buildup of fluid in the lungs, necrotizing fasciitis or flesh-eating disease, and it's also a common cause of food poisoning. And so you just want to have an idea of the types of diseases that are linked to Staph aureus. Now, when you're studying MSA, you want to know the purpose, right? And the purpose is to presumptively identify Staphylococcus aureus and select for the growth of salt-tolerant bacteria. You want to know the general ingredients. Again, the ones that really matter are the selective ingredients and the differential ingredients. So beef extract and peptones, you don't have to memorize that those are there for general growth. But you should understand in general that that's what those types of ingredients are used for. But what you do want to know is you do want to know that sodium chloride is included in the media, and it's at a high concentration. It's 7.5%. Inside the cell is approximately 0.85%. So almost 10 times higher concentration of sodium chloride is included in the media. And that's because that ingredient is selective for osmotolerant. Skin bacteria, remember, is going to be osmotolerant. So it's trying to allow for those skin type bacteria to grow, because remember, we're trying to identify Staph aureus, and Staph aureus should be osmotolerant. So we have high salt, which is selective for osmotolerant organisms. Our D-mannitol, that's our sugar. It's there for fermentation to produce acids. And again, this is going to be our differential ingredient, because remember from our table, Staph aureus can ferment it, Staph epidermidis does not. And so we'll be able to look at the agar to see if it's presumptively Staph aureus. And then lastly, we have our fennel red. And fennel red, we all know, is our pH indicator. It's pink when alkaline and yellow when acidic. So for studying for the quiz, you don't have to know the procedure for this, okay? You want to have a general idea, which is that you swabbed your nasal passages. That's where you would have swabbed if you had done this. And you would have rotated that on the plate, and then you would have done a standard streak plate. So you do want to know what the different colors on the agar tell you. So again, if we see growth on MSA, growth at all tells us osmotolerant. So any growth that's on the plate is going to be our osmotolerant. Again, selective ingredients would be our sodium chloride. We're selecting for osmotolerant bacteria. Differential is going to be our D-mannitol, because again, Staph aureus will ferment it, Staph epidermidis does not. We have our fennel red, which is our pH indicator. So you want to understand what these different colors tell you. So if the agar turns yellow, now I'm not talking about the bacteria itself, but the agar itself is yellow, that tells us that the pH is acidic, because remember that phenyl red turns yellow and acidic. We get acids produced if bacteria metabolize the mannitol. And if they've metabolized the mannitol and produce acids, it's going to lower the pH, causing the phenyl red to turn yellow. That is presumptive for Staph aureus. It's possible that it could be another type of skin type bacteria called Micrococcus, which is non-pathogenic. And so if you get yellow agar, you would want to follow up with a coagulase test, because notice that the MSA is only presumptive, meaning we can say with about 95% confidence that it's likely Staph aureus, but to confirm it, we would do coagulase. The coagulase would be our confirmatory test, because remember, coagulase is a virulence factor. Staph aureus produces it, the others do not. Red or pink agar, right? So if the agar is pink, that tells us that it's an alkaline pH. Bacteria did not metabolize the mannitol, but instead used peptones as a food source, right? And products of peptone metabolism are going to be alkaline. They're going to raise the pH, and the agar is going to be pink. It is osmotolerant because it grew, but it did not ferment the mannitol. And so that is likely Staph epidermidis, right? It's likely Staph epidermidis because if you tested your nasal passage or you tested your skin, you're likely going to have Staph epidermidis there. And so those pink colonies are possibly Staph epidermidis. This would be a negative result in an MSA because, again, we're trying to presumptively identify Staph aureus. Staph aureus should be able to ferment the mannitol. If we did this and we got no growth, that would be no osmotolerant bacteria. So that again is not Staph aureus. Now, as a control, if you were doing this, if I swab my nasal passage and I streak on an MSA plate and I get no growth on my plate, can I say that there's no bacteria in my nasal passage? Answer is no, right? Because the only thing that can grow on that plate is bacteria that's osmotolerant. So as a control, I could also take a swab and swab it on a nutrient agar plate, because nutrient agar is going to allow lots of different types of bacteria to grow. There aren't selective ingredients. And so if I swab my nasal passage and I put the swab on nutrient agar and I see no growth, then there's no bacteria. But if I see no bacteria on the MSA, my only conclusion is that there's no osmotolerant bacteria. I can't say no bacteria. I just can say there's no osmotolerant bacteria. And so again, you want to have watched these videos to go through this more slowly. I'm just going through this quickly just to review and to help you know what you should know for your quiz. So you want to know, what does yellow agar tell you? What does pink agar tell you? What organisms is that presumptive? What did they use as a food source, etc. And so that's what you want to review for the MSA. So the coagulase test is to test whether bacteria produce coagulase like Staph aureus. And again, this would be our confirmatory test after doing an MSA. And so we would inoculate a tube that has rabbit plasma in it. And so rabbit plasma, it's just like we have plasma. The plasma is the liquid part of the blood, and it's what contains the clotting factors. And so in that rabbit plasma is going to be our substrate for this test, which is fibrinogen. So fibrinogen is a clotting factor. Now, if there's bacteria in the tube and the bacteria produces coagulase, this is going to be an exoenzyme because the bacteria needs to secrete the enzyme outside of its cell to produce the clot around it. And so this is why this enzyme is an exoenzyme. And so if the bacteria is present and they are coagulase positive, they're going to secrete this coagulase. They're going to produce the product, which is the fibrin clot, and that's going to be a solid. So that if I look at these tubes, these coagulase tests, if at the end, if it's solid, that's positive. That's confirmatory for Staph aureus. If it is negative, that means that it's coagulase negative and it's not going to be Staph aureus, or at least not a pathogenic Staph aureus because it lacks coagulase. And so just to kind of tell you how you did this, again, you don't need to memorize this for the test, but the idea is that when you did this, if you had done the MSA plate and you had from your nasal passage yellow colonies, meaning the agar turned yellow, then to confirm that you actually carry Staph aureus, you would take your culture, your yellow colony that you found on your MSA plate, and you would use that colony and you would inoculate a rabbit plasma tube. And then after the 48 hours, we would look at and see if it turned solid. If it turned solid, that would be confirmatory that the yellow agar, the colonies that form the yellow agar, were in fact Staph aureus. And so the coagulase would be our confirmatory test, because Staph aureus and Staph epidermidis, as well as other types of skin bacteria, will grow on MSA. But we're looking for Staph aureus specifically. So in terms of the throat culture, you don't have to memorize all the different types of plates that were shown as an example. Those were just to give you some example. But you do want to understand about blood hemolysis, the breakdown of red blood cells. And we'll talk about that in a minute. And so just a reminder for the throat cultures, you would have been swabbing the back part of your throat where your tonsils are. If you don't have tonsils, I don't have tonsils, you would just swab where the tonsils should be. And you would want to avoid this little thing that hangs down, the uvula, so that you're swabbing the back of the throat. And so let's look at types of hemolysis that you might see on a blood agar. We also talked about, and these are all going to kind of go together, and that is the CAMP test. And the CAMP test was done in conjunction with this. And the purpose of the CAMP test was to identify and confirm the presence of group B strep. So specifically, Strep agalacti. Strep agalacti, it's important to know if you're a carrier. There's a question in your question set that talks about a beta hemolytic strep, and to know why it's important to know if you are a carrier. And for that one, you're talking about Strep agalacti, because knowing that you're a carrier if you're female and you're pregnant, knowing that you're a carrier of Streptococcus agalacti, you would be given antibiotics during delivery to try and prevent transmission to a newborn, a neonate, because if not, and this bacteria, or as the baby passes out the vaginal canal, if the baby ingests this Strep agalacti because it's in the woman's vaginal area, it can cause a serious infection in newborns. And so one way we can test for group B strep is by doing a CAMP test, and I'll describe this in a minute. And so there were 4 other bacteria that we were testing: Streptococcus pyogenes, which causes strep throat; Streptococcus mutans, which causes cavities; Streptococcus enterococcus, which is now renamed to be Enterococcus faecalis. Faecalis should tell you feces. It's in the GI tract. And then Streptococcus pneumoniae, which causes pneumonia. And so let's talk about this. So, what you want to be aware is that when we talked about the setup for this, you have to realize that this was more, excuse me, this was more than just the camp test. We did multiple tests on the same plate. You take your loop and you basically you would make a lawn over about approximately half of your plate. So you would streak your unknown strep species about half of your plate, 2 broad streaks down. You leave a short gap, and then we also would streak Staph aureus on the bottom of the plate. This part here, the part where you're doing the lawn and the streaks down and the Staph aureus, that is the camp test. On the same plate, we added 2 other tests. These are not part of the camp test. So we did an optogen sensitivity and a bacitracin sensitivity, which is essentially a Kirby-Bauer. It's a disc diffusion method to see if the bacteria is sensitive to either of these 2 drugs, because those sensitivities would help us identify different Streptococcus species. So be aware, these are not part of the camp test. You don't want to say that you would expect to see, if it's optogen positive, that it's camp positive. No, it doesn't work that way. They're different tests. We just conserved by putting it all on one plate. And so just be aware of that. So the other thing that we talked about was bile esculin agar, because again, this goes back to identifying different Streptococcus species. And so the purpose of our bile esculin agar, select for the growth of Enterococcus for presumptive identification. So specifically for bile esculin, we're trying to presumptively identify Enterococcus faecalis. That's what we're presumptively identifying. So presumptive for E. faecalis specifically. You want to know the ingredients in this media. So review this slide, review these notes. You have to know these ingredients. So we have peptones. Peptones are there for general growth. We have oxgal, or our bile salts. This is used to inhibit gram-positive. If you think back to when we did our coliform testing, right, we had BGLB, which had bile salts. And if we were trying to grow coliforms are gram-negative, right? So in our BGLB broth, we had it there to inhibit gram-positive. In this test, it takes it a step further because the bile salts will inhibit gram-positive except Enterococcus. So this ingredient is selective. We have our sodium azide. Sodium azide is going to inhibit gram-negative. So again, this is selective. So notice that we have 2 selective ingredients. One inhibits gram-negative. One inhibits gram-positive except Enterococcus. And so what that tells you is that anything that grows on bile esculin agar must be an Enterococcus. It's the only thing that can grow. We have our esculin. Esculin has glucose in it, and it's a special food source. It's differential for E. faecalis, meaning other Enterococcuses can grow on bile esculin agar, but they don't metabolize esculin. E. faecalis does. Ferric citrate is the reagent that's in the media, so we don't add it after, it's actually in the media, and it turns brown with esculin. And esculin is a product of esculin digestion. And so what that means is that if the organism uses the esculin, if it metabolizes the esculin and it produces esculin, that esculin is going to interact with the ferric citrate and it's going to turn brown. And so let's look at that. So this slide you want to review. Okay, so esculin is going to be your substrate. It's your food source. Your product is going to be glucose. So that's the reason that organisms can use esculin as a food source. It produces glucose, which is used for food. And the other product is going to be the esculin. And this esculin is what is going to react with the ferric citrate. And it's going to react with the ferric citrate in the media, and it causes that dark brown color. So if we see growth on bile esculin agar, but it's not brown, so growth but not brown, it must be an enterococcus because it's the only type of bacteria that can grow, but it's not E. faecalis. That's what that tells us. If it's growth and it turns the agar brown, that is presumptive E. faecalis. Because again, E. faecalis will use the differential ingredient, which is the esculin. It will metabolize the esculin. It will produce esculidin. Esculidin will interact with the iron, and it causes the dark brown color. The way I remember this one, brown poop, right? E. faecalis, faecalis feces. Feces is the poop colored one. So the brown is the enterococcus faecalis. And so that would be the growth, and brown would be positive for presumptive E. faecalis. So in terms of the blood agar, you want to know what the M protein is. And so the M protein was identified by Rebecca Lansfield, and it has antiphagocytic properties. It's like Velcro. It allows the bacteria to stick to surfaces. And when the bacteria sticks, it makes it difficult for the white blood cells to engulf it and to do phagocytosis. You want to review this, and that is, what are the types of hemolysis? So beta hemolysis, you're going to get clear zones around the colony. It almost looks like the agar is a little bit yellow. It doesn't turn the agar yellow. The yellow just is what appears as the red blood cells get broken down. And so the beta hemolysis is the complete destruction of red blood cells. This will be the case for both group A and group B strep, which we'll review in a minute. Alpha hemolysis. This is going to be the partial destruction of red blood cells. And so you get this green zone around the colonies. And that's because the red blood cells, the hemoglobin, gets broken down into something called methemoglobin. And it gives the agar this kind of greenish hint because the hemoglobin is not fully digested. And so of the 5 strep that we test, Strep pneumoniae would be the one that would be alpha hemolytic. Gamma hemolysis, that's what this little symbol here, this is a lowercase Greek gamma. Gamma hemolysis is where we get no damage to the red blood cells. There's no change in the agar, so the bacteria will grow, but it does not break down the red blood cells. So there's no damage to the red blood cells, there's no change to the agar. And of the 5 bacteria that we test, Strep mutans and group D strep would be our examples. So you want to review this slide, and just for all of the slides about the diseases that go with these different organisms, you want to have a general understanding of the types of diseases that they cause. And so strep pyogenes, right? Pyo refers to pus, so they're pus forming. And so the diseases that are associated with strep pyogenes would be strep throat or pharyngitis, scarlet fever. If it becomes systemic, it can lead to autoimmune diseases like rheumatic fever, glomerulonephritis, so damage to the kidneys. It's the most common cause of death of most common cause of necrotizing fasciitis or flesh-eating disease. It also can cause toxic shock syndrome. So you just want to kind of understand what diseases are associated with strep pyogenes. But the biggest thing is that the strep pyogenes is a type of group A strep. It's categorized as being group A strep, and group A strep is beta hemolytic. Strep agalacti is part of group B strep. Group B strep is also beta hemolytic. Strep agalacti may be normal flora in the GI tract, and Some females, about 25% of females, are vaginal or anal carriers. And so this is the one, so knowing that you carry strep pyogenes is not as critical, right? Because only 5% of people carry strep pyogenes as part of their normal flora. And oftentimes, if it's there, it might not be problematic. It's not something you necessarily need to know. However, knowing if you're a carrier of Streptococcus agalacti, this is the one that you need to know if you're a carrier, especially and only really if you're female and you are pregnant. Because again, the group that's at risk for problems with Strep agalacti would be neonates, newborns. And that's because if the woman carries Strep agalacti or group B strep in the vagina, if she has that as part of her normal flora, and the baby comes out during vaginal delivery, if the baby ingests this, it can lead to pneumonia, sepsis, and meningitis. And so again, knowing that the woman is a carrier of Strep agalacti, you can give a prophylactic antibiotic during labor to try and prevent transmission to the baby so that we don't get those complications in the neonate. And so this is the one that you would want to know if you are a carrier of Strep agalacti. Group C strep we're not going to talk about because Group C strep is related to animals. And so we're going to skip over Group C. Group D strep is going to be gamma hemolytic, which remember means no digestion of red blood cells. We have our Enterococcus faecalis. Again, it used to be called Streptococcus enterococcus and it was renamed. Enterococcus faecalis is part of normal flora in the gut. 100% of people carry it as part of their gut. Problem is when it gets out of the gut. So a gunshot wound, a stabbing, even something as simple as a colonoscopy, if they nick the colon and some of the bacteria spills into the peritoneal cavity, it can cause an infection. VRE, vancomycin-resistant enterococcus, a major nosocomial pathogen, meaning you go to the hospital for something else and you might leave with this type of antibiotic-resistant infection. And so again, you don't have to know all the details of this, but you do want to know that it's found normally in the gut and it becomes problematic if it gets out of the gut. And then our last two of our 5 that we would test would be our Streptococcus mutans, again, carried by 100% of people, and it's the cause of cavities. And so it's going to use sucrose, which is table sugar, to make dextran. And dextran is a large complex polysaccharide, and it causes this biofilm and plaque to form. Now, if you have a biofilm, right, so let's say you get that biofilm on your teeth from the bacteria growing there, and now you eat sugars, well, bacteria metabolize sugar and produce acids. The acids erode the enamel, and you end up with a cavity. And so this is a cause of cavities because it's bacteria found in your mouth, and it has the ability to form biofilms and to form that plaque on your teeth. Strep pneumoniae obviously causes pneumonia. It's the most common cause or most common form of pneumonia. There are other bacteria as well that cause pneumonia, but this is one of the more common. About 50% of people carry it. And so, again, you just want to have an idea of what diseases these bacteria cause. So, this slide I was just kind of showing you how do we identify which bacteria is which. And so in our CAMP test, again, the purpose is to identify and confirm the presence of group B strep, which is Strep agalacti. And so what you're looking for in the CAMP test, remember that you streak your Staph aureus. Staph aureus is also beta hemolytic. We have over here, remember I told you you're going to take about half of your plate and you're going to streak a lawn, and that is going to be your unknown Streptococcus species. And so if that Streptococcus species is group B strep, which is Streptococcus agallecti, this bacteria is producing a CAMP protein. This Staph aureus is producing that beta-hemolysin, which is able to break down the red blood cells. And so those two combined, right, you also have a broad streak here. This bacteria is producing this CAMP protein. This bacteria is producing the beta-hemolysin, and you get this enhanced synergy between that CAMP protein and the beta-hemolysin, and you get this synergistic clearing, this triangular shaped clearing, which is characteristic of a CAMP test. So when we do this test, we had 5 unknowns. Those were the 5 Streptococcus species that I talked about. And number 3 is the one that is CAMP positive. If we look at the plate for number 3, we would see this triangular clearing. And that tells you that it's CAMP positive. CAMP positive would be Strep agallecti. And so that's what you're looking for. You're looking at this synergy between Staph aureus and the CAMP protein that's produced by Strep agallecti. And so you get this extra triangular clearing here. Remember I said that on the same plate, right, on the same plate, you would have had discs in this lawn area, and we were doing an additional test with it. Those discs on the same plate were not part of the CAMP test. They were used to identify the other types of bacteria. Because of the 5, only Strep agallecti would be CAMP positive. How then would you identify the other 4? Well, because we have differential tests for the other ones. So one of the things that we did was we had a optogen disc. This is an antibiotic. It's referred to as the O disc. And we're looking for optogen sensitivity. And remember that for antibiotic sensitivity, you're looking for these zones of inhibition, this clearing. If we see this clearing around the disc, that means that the bacteria is sensitive to the optogen. And of our 5, the only one that is sensitive to the optogen would be our Strep pneumoniae. It would also be alpha hemolytic. And for unknown number, that would go with number 2. So number 2 would have been our Strep pneumoniae. We also had another disc on the plate, which is a bacitracin disc. Bacitracin is also referred to as the A disc. And of our 5 organisms, the only one that is sensitive to bacitracin, meaning it would have a zone of inhibition, would be our Strep pyogenes. It would be beta hemolytic because it's a group A strep, so it's beta hemolytic, and that would be organism number 1. And so this would be organism number 1. So 1 was pyogenes, 2 was pneumoniae, 3 is agallecti, which means we still need a way to differentiate between Strep mutans and our Enterococcus faecalis. And so we need one more test. And so these are the differential tests to identify these different Streptococcus species. The CAMP test is used to identify group B strep or Strep agallecti. Optogen sensitivity is used to identify Strep pneumoniae. Bacitracin sensitivity is used to identify Strep pyogenes. So how do we differentiate between our Enterococcus faecalis and our Strep mutans? Because they're both gamma hemolytic, so we can't look at that. So we need an additional way to differentiate those. And so the answer is you look at your bile esculin agar. You would also inoculate a bile esculin agar, and if it grows and it's brown, it's going to be E. faecalis. And so in our example, number 4 was the one that was positive for E. faecalis. On the blood agar, it would look, you would have gamma hemolysis, it would grow, but it would have no breakdown of red blood cells. And instead, you would have to then look at the bile esculin slant. We do it on a slant, not on a plate. And you would look at your bile esculin slant, and if it's brown, that is going to tell you that that organism is your E. faecalis. So number 4 would be your E. faecalis. So this slide is just summarizing your 5 unknown Streptococcus species. Put a star next to this, either this table or the next diagram. You must know this. You must know this. You must know this. So Strep pyogenes, you want to know its Lancefield classification. That's group A strep. You need to know that it is beta hemolytic. And you need to know that the differential test to identify Strep pyogenes is sensitivity to bacitracin. Strep agalacti would be your group B strep. It is beta hemolytic, and its differential test would be that it is CAMP positive. It has that triangular synergy between that strep and Staph aureus. E. faecalis is going to be our group D strep. It is going to be gamma hemolytic, which means it's not going to break down the red blood cells, and its differential test would be the bile esculin positive. I'm going to skip mutans for a minute. Streptococcus pneumoniae is a non-Lancefield, so it doesn't fall in this group A, B, C, or D strep. It's this other group. So Streptococcus pneumoniae is non-Lancefield. It is alpha hemolytic, which remember has a green hue on the blood agar, and it's sensitive to optogen. It's going to have clearing around the O disc. So for the strep mutans, how you would identify it in this test is it's a non-Lancefield. It is gamma hemolytic, so on the blood agar, it's not going to have clearing, and it's negative for everything else. So it's not sensitive to bacitracin. It would go right up to the bacitracin disc. It is not sensitive to optogen. It's going to go right up next to that optogen disc. It's not CAMP positive, and it's not bile esculin positive. So basically, it's negative to everything, which then by default would tell you that you're looking at the strep mutans. So I put this in here in 2 formats. So a table format, or my preferred, which is I am a very visual person, so I broke it down a little bit different. Alpha hemolysis, remember, is green, so I color-coded this green. If you see alpha hemolysis on a blood agar plate, the next thing you're looking for is optogen sensitivity. And if you see alpha hemolysis and you see optogen sensitivity, you're looking at strep pneumoniae. If the organism is beta hemolytic, then the next step is you have to look at, is it sensitive to bacitracin, or is it CAMP positive? If it's sensitive to bacitracin, you're looking at Streptococcus pyogenes, group A strep. If it is CAMP positive, you're looking at Strep agalacti, which is group B strep. If it is gamma hemolytic, so it does not break down the red blood cells, the red blood cells are still red, which is why this is color-coded red, then you would look for bile esculin test. If it is bile esculin positive, you're looking at Enterococcus faecalis, which is your group D strep. If it's gamma hemolytic and it's negative for everything else, by default, it is strep mutans, which is our non-Lancefield. And so you must know this. The purpose of doing all of these other tests was to help us identify our unknown Streptococcus species. And so what you would have done in class is we would have given you one of those 5 bacteria, and you would have had to figure out which bacteria you and your partner had. And so this would be how you would do process of elimination to figure out which organism you were given. So the last part that I want to go over, and I'm going to review this very quickly, I'm not going to go through all the details, but I just want to review what you want to study for your fungi. So You want to understand what mycology is, so the study of fungi. Um, this is going to include your yeast, molds, and mushrooms. You want to know your characteristics, of all fungi. So review the list that I gave you about what are found in all fungi. So they're eukaryotic, right? Most are saprobes, etc. So go back and review the characteristics of fungi. You also want to be able to compare and contrast yeast and molds, right? And so yeast would be unicellular fungi, molds would be multicellular. When they grow on a plate, one of the things that's different is that the yeast look like bacteria. The colonies are very smooth, whereas the molds, the colonies have kind of a velvety or fuzzy appearance. You want to know that the molds are filamentous, right? They're thread-like. They have hyphae, which are filaments that are used for nutrient absorption. The molds reproduce using spores, either asexually or sexually, and we talk about examples of this. Yeast often reproduce by budding, which is an asymmetrical division. And so you want to compare and contrast yeast and mold, so the characteristics for each. You want to know an example of a medically important yeast and what disease that causes. So again, it causes candidiasis, which is a yeast infection. It can cause a yeast infection in the vaginal area or the mouth. And if it's in the mouth, again, it's referred to as thrush. You want to know that it can become systemic. So if it becomes systemic and goes through the blood, that can be very fatal for patients. You want to know which group of individuals are susceptible for Candida albicans infections. Again, some people you'll hear called Candida albicans. It totally depends on who you hear talk about it. But you want to know which group of patients are most at risk for this type of yeast infection. You want to know an example of a non-pathogenic yeast. And your non-pathogenic yeast would be Saccharomyces cerevisiae, which is your baker's or brewer's yeast. Its name means sugar fungus that makes beer. And so this organism does fermentation to produce carbon dioxide gas and ethanol. And so that's alcohol. You want to know that if you were to look under the microscope and see something like this, this is going to be the yeast, not the mold. And again, when Saccharomyces cerevisiae is growing on a plate, it's going to have an appearance that resembles bacteria. It's very smooth and it looks like bacteria as opposed to molds, which are more fuzzy. This is just another example of looking at Saccharomyces cerevisiae. Again, you can see it's these individual unicellular organisms. That's going to be different than the structures you see for molds. And so this would be your example of a yeast. You want to know the 3 types of molds that we talk about. So we have our Rhizopus stolonifer, which is our black bread mold. You want to know that their hyphae are asepty, which means that there's no septum, and that the hyphae end up being what's called coencetic. They have multinucleated cells, so there's no wall separating the nuclei. You want to know what hyphae and rhizoids are used for. You want to know that in the case of Rhizopus, the aerial hyphae is referred to as a sporangiophore. Phore tells you aerial hyphae. And it's called a sporangiophore because the type of spores that are on it are called sporangiospores, which you're going to see in a minute. So you want to know what types of spores that Rhizopus uses. So it's going to use, the structure here is called the sporangium. And within the sporangium are a type of spores that are called sporangiospores. those are used for asexual reproduction. And you know that because there's only one hyphae going to the structure. There's no partner. On the flip side, if you see a fungal spore structure like this is called a zygospore. And the zygospore is used for sexual reproduction. You can see two hyphae coming together. We have this positive strain and this negative strain coming together, and they're reproducing sexually. So you want to know the names of these two types of spores, spore structures, and then which one is sexual and which one is asexual. Penicillium notatum. Okay, Penicillium notatum. This is your medically important mold, right? The big thing about this one is that it's used to make penicillin antibiotic. It's also used to make blue cheese and brie cheese. You want to know that this organism would have a septi hyphae. So again, it has the septum that separate the nuclei. It has its aerial hyphae is called the conidiophore. Phore again tells you aerial hyphae. It has these phyllides, which are these structures that hold these spores. These spores are not encapsulated like rhizopus, so these are naked spores, and they're referred to as conidiospores. Conidiospores are again used for asexual reproduction. Notice it's coming up from one hyphae. There's no partner involved. So these would be asexual reproductive spores as well. So you want to know that they're called conidiospores and that they're used for asexual reproduction. You want to know why fungi are grown on Sauerbrood's dextrose agar. And that is because the low pH of between 5 and 6 encourages fungi to grow and not bacteria. In addition to knowing what penicillium looks like under the microscope, you should also be able to identify it by plate. And by plate, you're going to see it being the spores are going to be green and the hyphae are going to be white. And so this is what Penicillium notatum would look like growing on a plate. You want to know Aspergillus niger. So this is a medically important black mold. It causes a lung infection, which is referred to as aspergillosis. It's used to make soy sauce and soy paste. It has septi hyphae. It has a conidiophore and conidiospores, just like penicillium. So these are naked spores. Ours look more like this. So you can see this ball structure. Again, it kind of looks like aspergillus, but the difference, it looks like rhizopus. The difference though is that rhizopus is going to be smooth because they're encapsulated, whereas these spores are naked. Again, they're asexual. There's only one hyphae here. They're asexual, and they're conidiospores because they are naked spores. Growing on a plate, you would need to identify. So black, so think of Niger means black. It has black spores and yellow hyphae. And so that's how you identify that. You don't, for this, have to memorize how to do the fungal plates. So you don't have to memorize that. And so again, you want to just review the pictures of the fungi and be able to identify them by plate and under the microscope. So this is what rhizopus would look like, the black bread mold on a plate. Here is it on a slide culture. Again, the key distinction here is that the spore structure, the sporangium, is smooth because the spores are encapsulated. These are sporangiospores. They're used for asexual reproduction. So again, this is a tape mount. So not a slide culture now, but a tape mount looking at rhizopus. And again, you can see that the spores are encapsulated, right? And so we have our sporangiospores inside this capsule structure. If we look at this one, these are going to be your zygospores, because here's our structure with our zygospore, and it has two hyphae coming in, which is used for sexual reproduction. If we move on to Aspergillus, you need to identify it on the plate. Again, Aspergillus niger, black are the spores, yellow are the hyphae. You want to identify it on the slide cultures. So again, they're like little balls, but the spores are naked. They use kinetiospores asexually. Here it is for a tape mount. Same thing. You can see the naked spores in this case, the kinetiospores. Again, we have one hyphae coming to it, so it's asexual. And then we have our penicillium. And so again, it's going to be green spores and white hyphae, so you need to identify it on the plate. These plates are easy to identify. They look very different, so it should not be hard to mix those up. Penicillium are the little fingers. They have the kinetiospores, the naked spores that are coming up. Here it is as a tape mount, same thing. So here are the phyllides, and then these are the kinetiospores. And so this is what penicillium would look like for a tape mount. So again, for these fungi, you should identify them by their plate. You should be able to identify them, what they look like in their tape mount, as well as what they look like in their slide culture. And so those are the things that you need to know for the fungi.